Chidima, how are you? Oh, bear with me. How are we doing? How's things? I am good, thank you. I am good. I literally have just finished today. Tonight it's gone live right now. My Patterns of Inheritance study guide. It has taken me so long. It's ridiculous how long it's taken me, and it's finally up. And I'm so happy that it is up and it is there. So people can purchase it now if they want to. Oh, I'm sorry to hear that. That's not good. That's not great. It's really hard. What is it that's is it the topic areas, or is it just everything in general? What's going on? Actually, fine. It's just you cussing everyone with injuries. Oh, I know. Do you know it is really hard, because this is the thing. When you start comparing yourself to someone else, you then start feeling like it's just you. You're the one who's not getting the good news. All these other people are getting all these interviews, options, things like that, and it's so tough. It's so tough. But keep believing that it will come. Hi, Safi, how are you? I'm just closing down all my new diagrams I've been making so I don't lose them. Hi, guys, if you're watching me on YouTube, please do, if you have topic areas you want to cover, ask away. I can't get TikTok to display my stuff like it did before. Keep believing. I know it's hard when you've had a rejection. If you're like me, your rejection sensitivity goes through the roof, then it's so so destroying when you just get rejection after rejection. But do believe that things will happen for a good reason when they happen. And I hate saying that sometimes because I'm like, well, what about the bad things that are happening? Were they meant to happen for a reason? But actually, what you have to realize is that just because maybe the ones that you thought were the right place for you, and you applied to them, maybe they're not the right place, and maybe there's some reason for that. As I said, for those of you who are just coming across for the first time, I didn't go to university until I was 20 because I just wasn't ready. I didn't believe in myself. I didn't feel like I could do what was needed to do, what I needed to do to be able to be successful. I didn't believe in myself. And when I compared myself to everybody else, they were all getting what they wanted, what they needed. And for me, it just seemed such like a unachievable thing. So I would say try and keep going, try and keep your head up. But I would definitely say that it will get better. Right now, it's dark, it's horrible, it's cold, it's wet. Everyone's getting like the cold, the flu, etc. It's a tough time of year, so try to just keep your head set on it's the end goal of where you want to go. You will get there, but you're going to have to work for it. So yeah, it is tough. It is tough. I feel for you. I'm glad I'm not going through it, to be honest. I'm trying everything I can do to help you, but yeah, unfortunately, you've got to go through the process. There's no shortcuts. There really is no shortcuts. Hey, did you check out the study guides I sent you? Muscles, absolutely. Let's do some muscles tonight, since you have asked so nicely. Where have you got up to with muscles? Nice. I'm glad you like the module one. Honestly, I'm so excited about the patterns of inheritance. I've spent such a long time on this one with the diagrams and stuff, and it's so silly. but I've wanted it. I mean, it's such a tough, dry topic area as well. We just started. I understand the difference between the 3 different types. Okay, but you haven't, I'm guessing, done the slide and filament theory yet. Is that where you're at? I'm just actinizing the different bands and zones make no sense to you. Right, okay. Absolutely fine. Let's go through that. Animal response, what do you think there? Where was it? I have like all these folders about where all my different stuff is. So then to bring it across to here. Yeah, if you come to YouTube, you'll be able to see the actual screen. That'll be easier. I don't know what is going on. I need to have a play with TikTok again because I had it and then I don't know what's happened. It's not allowing me to display the same thing. Hi. As what I'm displaying on my YouTube channel. I don't know if it's because I'm using OBS that it's decided that YouTube has priority. But yeah. So, right. Okay. Let's do a bit on the muscles. If you're just joining me, I'm going to go through the muscles topic area. If you have questions, please do. Oh, really? Yeah, it's an interesting one. So, do you know, it's why I've kind of got both of them going because I'm trying to find this one. It saves it, and you get to see the screen. Whereas when it's on TikTok, you don't obviously get to see the screen. It just won't work. I don't know why. But right, okay. So you said you've already done the 3 different types. So just to recap, when you're doing those and you feel confident, just always go back and just double check it. Do you know what the smooth muscle looks like? Do you know what the cardiac muscle looks like? Do you know what the skeletal muscle looks like? So this was a diagram that they actually gave in one of the questions, one of the years, where they wanted you, hey Slimus, they wanted you to actually be able to distinguish between the different types. So knowing non-striated, fibers are spindle shaped and uninucleated. So uninucleated, 1 nucleus. They're used for slow contractions, long contraction length. And then it did come up as a 6 marker. Striated muscle, branch fibers, uninucleated, striated muscle. Again, if we're looking at skeletal, fibers are tubular and multinucleated. Now, what's really interesting, so you've literally said exactly what people have been telling you in the fact that, oh, it came up in 2025. You'd be surprised how many students will go, right, because it came up last year as a 6 marker, I'm not going to revise that topic area because it's not going to come up. However, what I would say to you is they will ask questions, but they'll ask questions in different ways. So they won't give you a 6 marker. Absolutely, it won't be a 6 marker again. However, what they will do is they will give you some questions which are related to the knowledge they didn't assess. So don't miss it out on your revision. Obviously, you will be assessed on everything, and it just depends how they assess it and where they pick and choose where they're going to go. So one of the things that a lot of students got caught out in, I want to say 2023. Was it 2023? 2022 had a lot on the kidney, and 2023 had stuff on the kidney as well, but in a different way. So they were linking the kidneys to animal response, whereas in 2022, I think it was, they were more about the kidney structure and what it does. So yeah, they definitely come up again, and it's important that you're always looking at everything and finding what you don't know. 2023 in AQA. Oh, interesting. If it was 2023, then it's probably 2023 in OCR as well, because they ask very similar topic areas. It's quite fascinating to look at they ask them in different ways, but the papers are quite similar in their topic areas. Right, let's go through then the structure of the muscle. Why does this feel like it's an old one? I don't know why. This feels like it's old. Give me 1 second. I don't think I've got the right one up. Muscle contraction. Yeah, I haven't got the right one up. Whoever designed NXL, I would agree with you on that one. NXL, I did, how many years ago now? When I first came to do the tutoring online in 2022, when I went from being in the classroom to being here, yeah, I started tutoring someone on that one, and I was literally like, this is horrible. It was awful. I hated it. Hold on, let's go. Right, here we go. This is the better one. Right, so I've got to just move things around. Bear with me as I tweak this slightly. What have we got when it comes to muscles? Oh, can you also label the TEM photomicrograph as well? That really confused me. Yeah, of course. I know this isn't directly linked to the topic, but what is the best way to revise for biology? Biology is hard to revise for because it's so much content. So my advice to you when you're revising is to go for the topics you hate first, because actually every other topic area can come in at the same time. No, it's not GCSE, it's A-level. Excuse me. So yeah, so when you're looking at the content with there being so much, pick stuff that you hate, but as you're going through and revising the stuff that you hate, so for example, we're about to do muscle cells. If you're struggling with the muscle cells, as we're going through, I'm going to show you and tell you where the other topics come in. So you're actually not just revising muscles and animal response, you're also revising those other topic areas. If you hate all of it, what do you do? I would say if you hate all of it, pick the bit that you would absolutely detest coming up in your exam first. There'll always be something that's worse for you than another topic area. Even if you hate it all, there will be something that is harder than the rest. Okay, so muscles, muscle tissue, muscle fiber. So when we look at the muscle is an organ. An organ is made up of tissue. Tissue is made up of the cell. So the muscle fiber is the cell. So what you have to remember, that's going right the way back to the cell structure. We have a cell that is made up of a plasma membrane that acts as a barrier to the outside and the inside. So the sarcolemma is the plasma membrane for the muscle fiber. Now, within each muscle fiber, what you're going to find is these cylindrical structures called myofibrils. Now, they are what are made up of what you know as the contractile sarcomeres. So it's important to remember it's an organelle inside a cell. So the sarcolemma is a plasma membrane. The cylindrical organelle is your myofibrils. Why did I ever think GCSE biology was hard? I know, right? It's funny because every student comes to A-level biology being like, I got a 7, a 6, a 7, an 8, and 9 in my GCSE. I can do A-level biology. And then they get like C's to begin with, or E's and D's. And they're like, what is going on? It's hard and you have to work at it. It does look like A-level chemistry, but luckily enough, it's slightly easier, I would say. But then I have to say, I'm going to come to the contractile sarcomere. Don't worry, I'm going to lead up to that. I didn't do chemistry as a GCSE. I didn't do chemistry as an A-level. Because I went to school in Scotland, I got to choose and I chose biology. So I only ever did biology. So when I went to university, I didn't know any chemistry. I had to do chemistry for my degree and it was tough, but I managed to scrape through and get what I needed to. And when I went to Cambridge to train to be a teacher, it was a science teacher with GCSE up to physics and chemistry. So I can teach all three of the sciences up to GCSE, but I'm a biology specialist. But I've been on courses where I've had to learn the chemistry so that I could then teach it. And as I've been going through this, I've been learning A-level chemistry just enough to take you that little step further for the biology side of things, but then not too far. So if you start sprouting some chemistry at me, I will literally run for the hills because I'm like, I don't know. And I have a very good friend of mine who does OCR chemistry and we go for coffee every month. So I've still got a co-worker that I can meet up with, and I sometimes pick her brain. I'm like, so I'm teaching this in biology. Can you explain it a bit more? Give me the chemistry. Tell me that. And I want more. So yeah, it's really tough. Yeah, it was to do with the... Oh, thank you for the likes, by the way. Oh wow, you got a C in your membranes test, but you just got an A in the last test. That's really cool. And I was integrating. Amazing. Amazing. Right. Okay. Chidima, I'm back. Contractile sarcomere. So what is it? So we have the cylindrical organelle, and surrounding the cylindrical organelle, we have got what's known as a sarcoplasmic reticulum. So these red circles here, imagine that that's the top of the red circle here. Okay. So what I'm showing you is if I draw this like this. So if I draw, oh no, that's red. I don't want red. I need black. Let's try that again. If I draw this like that, so what I'm looking at here is I'm looking at this part here. Okay. Oh, hi. So yeah, so what you've got is the myofibril, okay, is a cylindrical organelle. This is my myofibril, and my myofibril is surrounded by a sarcoplasmic reticulum. A sarcoplasmic reticulum, remember you've got your smooth endoplasmic reticulum, you've got your rough endoplasmic reticulum. The sarcoplasmic reticulum is the one inside the muscle cell. Yes. Yes, it's like you. So we're looking at a transverse section, then we're looking at a longitudinal section. So what I'm trying to do is I'm trying to show you the side of things. Oh, Daryl, you got an A on the proteins and the nucleic acid test, and you use the notes. Amazing. You're year 12, so you don't need it now, but I've just finished the Patterns of Inheritance study guide, and it's now on my website, so I'm really excited about that. It's a long time it's taken me to do, but I'm really pleased for you. Well done. That's awesome. That's really great. Right, okay, with the sarcoplasmic reticulum, it stores calcium. Yes, Chidima, it does. Since it's similar to the smooth endoplasm, it would synthesize and package carbohydrates and lipids. So you've got to remember that it's got different roles in different cells. Every cell is differentiated to do the job of what it needs to do. So if it's a cell that needs to synthesize and package carbohydrates and lipids, then that's what the smooth endoplasmic reticulum does. However, we've got this sarcoplasmic reticulum, and its job is to store calcium, because calcium is required for the contraction of the muscle. So remember, it's not about that it only does this or it only does that. It's important that you're looking at the fact that it will do what it needs because it's differentiated to do the job that it's going to do. I've got to be careful because I'm going to lose my voice again, and I've only just barely got it back. Okay, so here is a electron micrograph showing you a picture of the muscle. So as you can see, I've got these different sections up here. So I like to actually visualize what it is I'm looking at. So if I'm looking at these three, what they've basically done is they've taken, for example, a slice through those three. Actually 2.5, but you can see that I've got 3 red circles here. I've got my 3 red sections up here. So that is a myofibril. So V is my myofibril. So then earlier I mentioned about a contractile sarcomere. So a contractile sarcomere is the bit that's going to contract, the bit that's going to shorten. It's just a bunch of lines. Oh, Chidima, I get that it's a bunch of lines, but hopefully what I'm about to show you will make it make sense. It looks like it's a bunch of lines, but actually if you know the structure and you break it down, it does actually make sense. So I could, if I jump ahead, I'll quite easily say, right, let me change colour so it makes it more obvious. If I were to show my Z line, there's my Z line. there's my Z line, there's my Z line, there's my Z line. Here's my M line, it goes in between my Z lines, and so therefore I have a sarcomere, I have my Z line to my Z line, and I have my M line in between. So I have jumped here, and I will cover more in a minute, but just so you know, yes, X would be one of the Z lines. Yes, it would. Darrow, to answer your question, the next study guide I'm going to do is probably when I get a moment to do manipulating genome, because that's quite a challenging one as well. Okay, so let's move on. If we have the part of the sarcomere that we need to understand is that there are two different filaments, okay? So a sarcomere is made up of these two different filaments, and the sliding filament theory is that these filaments are going to slide over each other. And so what we have to understand is that we have got what's known as the thick filament. So the thick filament is composed of a protein. It's a protein that was made through transcription and translation. So someone said to me, how is it that you revise the other topic areas? So right now I'm going to talk about the myosin, the thick filament, but in my head I'm going, okay, it's a protein. What do I know about my proteins? And if you've ever been tutored by me, you will know I'll then jump and tell you, right, give me what's the first level, what's the second level, what's the third level, what's the fourth level. So therefore, do you know the primary structure? Do you know the secondary structure? Do you know the tertiary structure? Do you know the quaternary structure? So starting to bring that knowledge in, that's a quick recap of protein. So we're doing the muscles, we're looking at a protein called myosin. So therefore we know that it must have a very specific shape to allow it to do its job. So it has been folded in the way which means that it can have specific areas such as a tail region. So here's my tail, and this is what forms this core section here. So these bits that stick out are called the heads. So the head has got an actin binding site. So it has a site on it which is complementary. Its shape is complementary to another protein that we're going to look at called actin. It also has an ATP binding site. So it has a site which is complementary to ATP, which means that it is going to allow ATP to bind, and it will then work its magic on the ATP, i.e. it will hydrolyze it down. So these are all things that you would be then linking into, oh, okay, so it's going to have an enzyme section because we're talking about protein. We talk about the thick filament being myosin, and it has this binding site, which will then be able to do the hydrolysis reaction. So therefore it will have an enzyme part of it. Then we talk about the hinge. So I like to use my hands to think about this. So if this is the tail and this is the head, my hinge region at my wrist works in the same way that it can do this and this, this and this. So it's a hinge, and because it's a hinge, it means it moves back and forth. And this is the key thing. I love that you're saying this, because that's what biology is about. It's about bringing in the year 12 content that you've been learning to see it's the bigger picture. Biology is all interlinked. You learnt the foundational skills in year 12, and now we're going to put them into context so that they make sense, so that you can then apply it to whichever direction they decide to go in the exam, because they'll never do it in exactly the same way. I love this. 7 people are picking in. Break the ice to start interaction. Please do come join me, and also come and join me on my other channel where you'll actually see what I'm doing as my board. Okay, so let's keep going. So we had our thick filament. The thick filament, we've got these proteins. We're now going to look at the thin filament. Oh, hold on 1 second. Because of my branding, I've got to move this down slightly. So my thin filament, you can see my thin filament here. So I have my pink circular spherical actin molecules. So actin is these two really long chains which are twisted around each other. I never really understood how ATP is used in the power stroking class. I think this will help. Yeah, hopefully it will help. So we'll see by the end. If it doesn't, then obviously go watch my video again and see if it helps you. But yeah, it's a hard one to get your head around. I used to always go into YouTube, find the video that I found really useful, and then send that link to my students and be like, watch this on YouTube. And here I am doing this on YouTube. Back to what I was talking about. Thin filament, actin. Now there's another two proteins we need to talk about. Troponin. Troponin is a molecule which can change shape. So the troponin is this molecule that's able to change shape, and it's going to change shape when the calcium ions are going to bind to it. So you will never be asked about whether it is fibrous or globular protein, Chidima. So don't stress yourself out on that one. They're going to give you ones that are really obvious, or they're going to tell you about what it is. So when we talk about the muscles, they're looking for you understanding the three different proteins. And if they want you to say whether it's fibrous or globular, they'll give you some information in the question about that. And the reason being is because they've specifically said in the specification which ones they want you to know as your example ones. So the tropomyosin, the key thing to remember is fibrous ones are insoluble. Globular ones are soluble. So that's one of the ways in which you would distinguish between whether it is insoluble. Is it made up of many hydrophobic amino acids, or does it have hydrophilic amino acids? Hi, do you do private tutoring? Yes, I do. If you go to my website, ukbiotutor.com, it also should be in the link, you will see a way to contact me. But yes, I do private one-to-one tuition, and I do group tuition as well. So does the shape of the troponin change shape because when calcium binds, tropomyosin moves? So when calcium binds to the troponin, that is going to change its shape, and that is going to move the tropomyosin out the way. So it's important to think about your wording and the order of your wording. So we've got a relaxed muscle, the tropomyosin, which I've shown in my blue diagram here. My tropomyosin is actually going to be lying across the actin and the myosin blocking. So if you imagine that this in my... Yeah, I do love saying that as well. So if you imagine that this is my actin and this is my myosin head, okay? So I have my actin, I have my myosin head. Now we want them to combine together. So because we want them to combine together, we need them to, we've got a myosin binding site, we've got an actin binding site. They're going to come together, they're going to bind, they're going to form cross bridges. And this is where I'm like, do I have a bit of tissue here, so I'm going to use this as my example. So here is my myosin. No, it's not. Here's my actin. And here is my myosin. So I'm going to come from above. So I want the myosin and the actin to come into contact. But the problem is there is something in the way. There's something blocking the actin and myosin from forming cross bridges. So that is the tropomyosin. So the tropomyosin, my tissue, is in the way. This is trying to form cross bridges, but it is being blocked. So what I need, it's going to be really weird, but imagine me, my mouth, is troponin. If the calcium binds to me, I can then move the tropomyosin out of the way. I wouldn't drop it. I would just move the tropomyosin out the way. So there's my actin, my myosin. When that comes together, the fact that that can come together, that has now formed cross bridges. So my tropomyosin was moved out the way, and the reason it was moved out the way, oh, I hit the wrong button there. The reason it was moved out the way is because, if you can see here in my diagram, my troponin has changed shape, and because it changes shape, it moves the tropomyosin into the grooves in between. So it's now exposing the actin. Thank you. This is how I do it. This is why I do these lives as well. So I do these sessions. Obviously, I do my group tuition sessions. I do my one-to-ones. But I do these live sessions as well because I know for a lot of you, you can't afford tuition. And so therefore, I want to give back and help, because I know when I was your age, I couldn't afford it. I still can't afford it if I was actually doing it the other way around. Because I want to help you. So the fact that I build the resources, the fact that I do this is because I want you to actually be able to understand it. Now, obviously, if you want to pay me, amazing, great. So in my group tuition sessions and in my one-to-one sessions, there's none of this chatting away about different stuff. It's very much you're here to learn. You've got me for an hour. I will literally scrutinize your understanding to figure out what you know and what you don't know. But with these sessions, I will go off in tangents. I will have a conversation. I will respond to whatever comes up, because that is just who I am, and this isn't a paid session, if that makes sense. But I will try and do what I can to help you. So if you do have questions, if you are unsure, asking them, typing them in here, I will respond to them. If you have questions on my other videos, I will respond to them. And the reason I do it this way is because that's where my skills come from. I've got 20 years experience of teaching this, and so I want to give back to you. When I went to Cambridge, and I trained at Cambridge, and they said to me, don't you ever go down the private route, Christine. This is it. You are trained as a Cambridge graduate. I mean, I've got a master's, I've got 2 postgrads from Cambridge. I have been trained at Cambridge, but I've got 20 years of experience as deputy of lower school, as a director of student progress. But my jobs have changed because of my girls, because I found a passion that I love. I love building these resources. If you told me I'd become a illustrator and a content creator, if you told me I would do all this, I'd be like, oh yeah, whatever, like that's not gonna happen. And now look at me, I want to keep doing this, and I will keep doing this. AA, I will respond once I finish my live, so I do try and respond as soon as I can. So thank you for the email. I will respond when I get a moment. Okay, so Chidima, you said to me, you were literally like, it just looks like a bunch of lines. It's a funny one. It does look like a bunch of lines, but if you know what those lines represent, you can then understand how the muscles contract. Now, when I'm in lessons, I'm literally like, oh yeah, check my muscles out. And I joke around because I've got no muscles. Like, I can barely lift myself up into a plank. When I do my Pilates class, which I just started, this is my new thing. When I do Pilates Reformer and I'm trying to lift myself up into a plank, I'm like, this is awful. My muscles have not done any of the exercise that I used to do before the girls came along. And so I'm having to rebuild the muscles and get them stronger. Now you do that through using them more. So going for a walk, that's about the most exercise I do. Now I do my Pilates. And so what I'm doing is I'm allowing for my muscles to contract. So how do they contract? Well, I mentioned earlier about my Z line to Z line. So your Z line to Z line, and then you have your M line in between. Now, the Z line to Z line is a sarcomere. So remember earlier I mentioned that they were contractile sarcomeres. So because they're contractile sarcomeres, you have got in my diagram 3 sarcomeres in my diagram that you can see. So if we were looking at a picture, if we go back to our original one, a second, I'll come back to that in a second. We go back to here, I can see 2.5 sarcomeres in this picture. In this diagram, I have got 3 sarcomeres. So what I need to understand is my thin filament, remember, actin, troponin, tropomyosin, they are attached and anchored into the Z line. My thick filament, the myosin head with its tails making the core, that thick filament is anchored into the M line. So this is the first thing I would do, is I would make sure that I know where's my thin filament, where's my thick filament. So because I know I've got my M line, I'd use the alphabet. I love using words, I love using letters to help me to remember my little tricks. M line, myosin, Z line, actin. If I know I've got one of them, the opposite must be in the other one. So in my M line, my myosin is attached. In my Z line, it's my actin. So coming back here, we therefore can see the Z line. So what is the sarcomere structurally? The sarcomere is from this Z line to this Z line. So all the way along where you have, there's an actin thin filament attached to a Z line. There's an actin filament attached to a Z line. So structurally, when you're talking about this, you're talking about this thin filament which is attached to this region that we call the Z line. That's all the level you need to know it to. You don't need to go into any more than that, because with A-level biology, they've simplified it as much as they can to make it make sense. It's the same with GCSE. They simplify it more to make it make sense. So what I try and do is I try and take you a little bit further to make it make sense, but not too far that it then becomes confusing. Yes, I'll also talk about the H zone, the A and the I band. Yeah, and I'll give you little tricks of how to remember it. But right now, the only thing that we're talking about is our sarcomere and how to identify it. So if I know where my sarcomere is, I can then identify where my M line is. And it's quite easily seen here. Because a striated muscle has got a striped appearance, we can see the stripes. So the Z line is the darker region here and here, and then it's surrounded by a light region. Now the light region is what you're referring to as the I band. The light band is the I band. The dark band is the A band. And then if you look in this section here, this is your H zone, but I'll explain that in a bit more detail. So here we have my light I band, my actin only. So my I band is this region here, which is the light region, and the Z line is in between. And then my dark band or A band is where there's myosin, so the red is representing the myosin, and then you've got my actin is overlapping slightly, just slightly, but the M line with my myosin coming out, because it's a thick filament, that gives that dark appearance. And therefore, that's how I get this dark or A band. Now, as I said before, as I've always said, I am all about using my little tricks to help me to be able to work it out. So dark has got an A in it. Therefore, the A band is the dark band, which is where the myosin is. The light band is the I band, and that is where the actin is. So you need to know about your dark band, you need to know about your light band, you need to know about where the actin is, you need to know where the myosin is. So that's the important part. But then, so if we look here, I've got my sarcomere. So from the Z line to the Z line is my sarcomere. If my sarcomere has shortened, so if I go back a second, see if I can actually do this in the right position. So here is to there. That is my sarcomere. If I'm going to contract my muscles, what happens is, and I've got to really think this one through. I'll maybe get the positioning wrong. Oh, I was so close. I was so close because I can see it on an animated slide. There we go. What's the region where there is only myosin? So the region that there's only myosin is the H zone. So when you've got a sarcomere that's going to get shorter, so hold on, let me go back. Can you see the H zone here? The I bands are smaller. Be careful, the I bands aren't closer, the I band is smaller. So if I have looked here, I've got my muscles contracting, my sarcomere has shortened, because you can see the difference between this one and this one. I should ask you to a quick question about my biology IB IA, because I'm so confused. Yes, give me a second, let me finish this, and then if you ask the question, type it in here. I'll then answer about the IA for you. If you can give me your subject area and then tell me what you're confused at, then I'll answer that question. So you've got a muscle contraction. Each of the sarcomeres gets shorter. Dark band does not change its shape. So the dark band hasn't changed. If you look at this diagram here, I love this one, where you can see the Z to Z has got shorter. My sarcomere has shortened. My light band has got smaller. My dark band hasn't changed. My dark band is exactly where it was. It has not changed its size. That makes sense? Right, so hold on 1 second. I'm going to do this. Right. Okay. How come the H zone is the same and I band is closer? That's so confusing. It's not. It is, but it's not. Okay, so Chidima, don't worry. Don't worry. This is something that comes up all the time. Okay, so remember I said that my hands. Actually, no, ignore that. Ignore my hands. I'm going to change it slightly. So before I mentioned about the myosin head and the actin, I'm going to change it slightly now. So imagine now using my arms, if I can actually fit in here. Imagine that my head, okay, this part of my body, this part is the M line. My elbows are the Z line, okay? So my elbows are the Z lines. The actin is my arms. Z line, my actin is the arms, okay? So what's going to happen is my arms are going to get closer to the M line. So my elbow is going to be pulled closer. I'll talk about how. It's unfortunate because I'm not big enough to do this, but my Z line to Z line is going, my elbows are going to be pulled closer to the M line, okay? But the actin doesn't go all the way forward, okay? It doesn't come all the way to the M line. There is a region, the H zone, where it won't actually cross. You are very welcome. You are very welcome, Alan, for this. Right, okay. So, hold on. Does that make sense, Chidima? Is that making sense to you now? Alyssa, I'm not sure if you're still here. If you are and you are still typing away, that's fine. Or send it to me via chat. Okay. I'm trying to think, is there anything else I want to see on this one? Is there any more questions about the light band, the dark band, the sarcomere, the thin filament or the thick filament? Okay. Enzyme, I'm doing temperature on invertase. In my method I use Benedict's, but I had to boil. However, my teacher said it makes no sense, although I got the bell curve results, as it would denature with boiling. So hold on, you were using the enzyme and looking at temperature on invertase, but that wouldn't or shouldn't have affected your results. I'm assuming that you use the enzyme with the invertase at different temperatures, so 5 temperatures, And then you use the Benedicts later on samples of what you've done. The enzyme doesn't matter anymore because you've already done, the enzyme's done its action. What you're looking at is how much of the reducing sugar is present based on using the Benedicts, and did you do a colorimeter and do a calibration curve. So it shouldn't just give you a bell curve. What it should have done is it should have given you the absorbance or transmission depending on what you did with the colorimeter. So I'm assuming you have done that. I'll come back and answer your question once you've done that. Why is the H zone the same? Okay. Let me think. No, don't apologize. Please don't apologize for these questions. This is what it's about. Being able to ask the question so you can figure it out. This is what I do best with, is being asked random questions and having to think. I love it. So don't apologize. Ask the questions. The questions are what helps you to understand it, because if you're not understanding it, I have to rethink how I teach it, which then makes it better for me. It's one of the reasons why they suggest you try and teach it to someone else, because if you can't teach it to someone else, you don't know it fully. So the M line, I'm going to try and give it to you in a different way. I'm trying to think of a way to make this make sense. It's an analogy that would actually make sense. Okay, so I don't know if this is going to make any sense at all. The M line is right. Hold on. Give me a second. I'm thinking on the spot here, so bear with me. This might work, it might not. It might be the biggest pile of rubbish. Right, okay. So imagine the M line is the anchored in table, okay? It's going to sound really weird. You're in class. In your class, you have got tables positioned like this. So there's a table in the middle, and then there is a table that comes all the way, tables that come all the way out, okay? Please bear with me, okay? I'm trying. Now imagine you are sitting, each person is sitting here. So there are, excuse me, 5 of you on each side of a table. So you're in pairs and you're on each side of the table, okay? Yes, yes. Use the analogy of it causing cramps if this were to happen. Kind of. I like that actual analogy. That does help sometimes. Hold on, I'm going to... crumbs. Let me just get rid of these. I'm being lazy because what I'm going to do. Go away. I want to do that. Give me a sec. Okay, so here we have our 2 rows of tables. Of course, my thing would have to be a bit dodgy in its shape. Never mind. Let's try and rotate you slightly. Kind of makes sense, right? So we've got these 2 rows of tables, okay? Now, what you have is crumbs. You are being given a rope that you can pull along, right? So this is the actin, which is attached to the Z line. So we're going to put one here. I'm going to put one down here. I'm going to put one up here. Nope, let's try that again. And we've got one on this side. I'm going to flip around. Okay, so Z line to Z line. So that's your sarcomere, okay? So we got our sarcomere from our Z line to our Z line. We've got our M line in between. I know it's a bit weird. Just give me a second. So this is my M line, which is a table that cannot move. Now, each of you sitting on your seat cannot move. However, you can stick out your hands, which is the head, okay? Now, if you stick out your head, i.e. your hands, if you stick out the myosin head, what's going to happen is this one can grab on to that actin if it is exposed. Now remember, I can do this. So I can put my hand out, I can grab onto the actin and I can swivel. Okay? So what's going to happen is that one's going to attach, and what's going to happen, yep, people are the myosin, sat in position on the chair. So the tables cannot move, okay? The M line doesn't move, the tables don't move, the people are stuck in their seat. The only thing that they can move are their hands. So they can grab on to the actin if there is no tropomyosin in the way, and they can move their arms by their hinge region, moving it closer. So what will happen is it will move closer, and then it'll move closer, then it'll move closer, 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 closer. But it will only move to the point at which that Z line has now reached the end of the myosin. Does that make sense? And hopefully if I've done it the right shape, there we go. My myosin table in the middle never gets overlapped by the actin. And so therefore I have this region here, the H zone, to be honest, I actually haven't looked into the M line in more detail on the structure side of things because I haven't needed to. But it is an interesting question. Have a think on that one. I am going to come back. 1 second. Right, it says it's nonsense. That's this question. Do you need to know the G0 phase? You should know about the G0 phase in mitosis. They could ask a question where it's gone out and can come back in, or it's gone out because it's differentiated. Yes, okay, yeah, so Queen, thank you. I really needed this clarification. You are very welcome. I hope that helps. The IA is a tough one. Also, is it bad that I described my results having a bell curve relationship since it had optimal and then denatured? So if you have described your data and your data has shown that as you've increased the temperature, the rate of the reaction has increased, and then the rate of the reaction has decreased due to the denaturation of the enzyme, that's not a problem. That's a description and an explanation. But I'm trying to get through, because you're talking, did you do the colorimetry? And did you get that absorption data? Grace, if you want to see, I'm on my YouTube channel. It won't let me display it on here for some reason. So do pop over. So the blue section, Chinema, come back to you. This is the H zone. No. The H zone is not myosin and actin. I go back up here. Your H zone is where there is the no overlap. So the overlap that you're talking about, so the overlap, if I color coordinate this, the overlap is this region now. That's the overlap, isn't it? So because that's the overlap, okay, so you used a colorimeter, you've got the absorbance. I've got to think this one through. So when you were looking at the absorbance, you saw that there was a high, it went up and then down, which determined that there was a higher concentration, I'm guessing, of the reducing sugar. Because there's a higher concentration of the reducing sugar, wherever it peaked, that is where you would assume that that would be the optimum. However, how are you linking that to the temperature? So you've gone temperatures on your x-axis, is that right? I'm trying to visualize this. Temperatures on your x-axis, and then your y-axis is the absorbance. Or did you do a calibration curve? Temperature is your x-axis, and then you've got your y-axis. So therefore, temperature's along here, absorbance is up here. No, so that shouldn't have done that. Okay, hold on, I'm trying to think this through. So did you, what's on your y-axis? Oh, 1 second, I'm going to verify I'm still here. Oh, it's after 10 o'clock. I think I'm wrong, don't listen to me. Don't put yourself down, Chidima. Temperature is the x-axis. What's the y-axis? Absorbance. Hold on, I've got to get my head around what you're doing here. So you've done temperature here, absorbance here. Have you used a calibration curve? Do you have known concentrations or not? Are you just going by absorbance? So if you're going by absorbance, I'm assuming you've got an increase and then a decrease, which would mean that there's more reducing sugar at the peak. So if there's more reducing sugar at the peak, how long did you do it for as well? What was your time frame? No calibration coverage. You told me I don't need it as I was going to calculate standard deviation. So that's a different thing. It's a funny one. I used to always say that you didn't need the calibration curve. So hold on. So if I've got this right, what you're saying to me is you've increased the temperature. You've therefore seen an increase in the absorbance and then a decrease in the absorbance after the what you would say is the optimum temperature. That's the optimum temperature. Because there's more reducing sugar, therefore there's a higher absorbance. And if you go above that, it's decreasing, therefore you're saying that the enzymes have denatured. Because the enzymes have denatured, they're therefore not able to catalyze the reaction. But you would want to have the reaction in a rate of the reaction. So if you're just describing and explaining the results, then yeah, that's absolutely fine. By saying it's increasing and decreasing. I did it for 10 minutes, 5-minute reaction, 5 minutes boiling. So you're not, the 5-minute boiling, when it hits 6 minutes, recorded the total. Hold on. 5 minutes of the reaction, then you took a sample and put that in with a Benedict's for 5 minutes with it boiled. Or you did it for 5 minutes at the temperature. You then boiled it to denature the enzymes. And then you took a sample at 11 minutes. That's a little bit... Okay. But you did the same to each of them. You boiled it for 5 minutes. So you did the reaction at, let's say, 20 degrees C for 5 minutes. Then you put it into 100 degrees C water bath for 5 minutes. And then you took your sample on the 6th minute. Then 1 minute later, so 1 minute later, you then took a sample after it had that condition. So the conditions that it had was 5 minutes within the temperature that you had, 5 minutes being boiled to denature the temperature. Then you took denature the enzyme. Then you took a sample and you did a Benedict's test on that sample to look at the reducing sugar. 5 minutes exposure, 5 minutes boiling, recorded when it hit 11 minutes on the stopwatch. Okay, so each of the conditions were given, each of them were given that. Therefore, you were taking the sample, and when you did that, you had your absorbance, and therefore, if there is a difference, you could use that and relate that back to the rate of the reaction on the temperature. So you should have seen an increase because there's more collisions, more temperature, more kinetic energy, more collisions. And then you get to that top peak. That's the optimum. So you're going to predict that's the optimum. However, I would say in year one for your IA, you need to think about improving it and you need to evaluate it. So you need to calculate standard deviation so that you can work out how variable your data is. You then need to discuss the fact that actually you would want to redo it if you haven't in a small increments between the optimum temperature so you can figure out what the actual optimum temperature was. The fact that you did the 5 minutes and then 5 minutes and then the 1 minute, I would say is also a question that you would want to evaluate. Why did you do it that way? Why did you not just take the sample straight after the 5 minutes? So put it in the temperature at 5 minutes, take a sample and put that straight into Benedict's. Why did you then denature? That would be my question. So the rate of the reaction would be the quantity that's been collected or measured over time, which is why I was asking about the time. So it's an interesting one. I'm trying to get my head around your one. She had a question in your mock today and it said, what are two features of stem cells that, where is it gone? Cannot be viewed under a light microscope that distinguish them from specialized cells. They have the capacity to undergo mitotic division. Specialized cells still have a nucleus which contains all the genetic information, and so a stem cell would have that as well. You wouldn't be able to view that under the light microscope because the light microscope doesn't have a high enough resolution. Okay, yeah, in your practical, you waited for a change, then stopped the timer, then did 1 over time. Yeah, because you're looking at the rate. Yeah, I said that for one feature, another feature. I had no idea. Right, hold on. I'm trying to think this through. I'm also still trying to answer the other question. 1 second. So what they're asking for you in the question that they're actually asking is about, do you know what stem cells are and do you know what they can do? So a stem cell has the capacity to go through mitotic division, whereas a differentiated cell doesn't. So if you can see a cell that has been visible in any of the stages, prophase, metaphase, anaphase, telophase, if it's in any of those stages of mitotic division, you know it had to have been a stem cell before, because a differentiated cell would not be able to do that. That would potentially be my other one. Because for Benedict's to occur, we need to boil it to see. Yeah, you need to, oh, but hold on a second. You boiling the Benedict's is so that the Benedict's reacts. You boiling it for the color change should have been straight after the 5 minutes. So you've actually done 5 minutes of the reaction. You've then taken a sample from that and put that into Benedict's. That would be what I'd be questioning. So basically I'm asking you, did you put your sample in a water bath at 20 degrees C and then take that and put that into a new water bath that's at 100 degrees? So did you basically do the same practical? Wow, my brain's getting slow now. Did you take your test tube, which had the enzyme and the sugars, did you take that into 100 degrees C, or did you take a sample and add it to Benedict's? And then when you added it to Benedict's, did that then, so that was what the 5 minutes were, but only a sample of it, not the whole thing. Because if you're just doing a sample, then it was nothing about the denaturing of the enzyme. Oh, well, passive inheritance, I would agree, is the hardest topic area. Check out my new study guide. I've literally just put it on my website today, tonight, before I did this session. I have basically spent, I'd say it's been the last month, any extra time that I've had to build this study guide to try and make it more accessible for you, because I agree. It is really difficult. You put the whole test tube in. Okay, so it's okay, don't worry about it, because the one thing about the IEA for the IB is really good is that you then actually talk about that in your evaluation. So that would give you points towards the evaluation, because you're thinking about, actually, if I'm going to do this again, if I'm going to improve this investigation, what would I do? And what you should do for improving it is that you should have actually only taken a sample rather than taking the whole test tube. The reason being is because the enzyme would have still been able to catalyze the reaction, so your timing's a bit off. However, the fact that every single one of your conditions and every single one of your temperatures were given the same conditions, that shouldn't hinder you too much as long as you write about it. So don't worry too much. Put that in your evaluation section. So you know how you've got your conclusion and evaluation section? That would be where you talk about the improvements. But don't forget to talk about your data and about your standard deviations, how close, and do a statistical test as well. So you would probably do a correlation coefficient for that. Finishes patterns is so hard. Wow, you've done so much. My skill is so behind, and we're finishing in May. I have to say, different schools will do it in different ways. Don't stress out. I wouldn't, when I go into schools to support them, if I was teaching, I obviously this last year I haven't gone into the school, I've just been doing it all online, but I don't finish until the end of April. Normally the first week in May, I give myself that leeway of doing it, and I run an intense course where I've taught the whole A-level in 1 year as well, and I still get up to that point. So just be aware that you can do it. Your teachers will do things in different ways. Aiming to finish by the end of December. Wow, silly, that's I'm not going to say anything on that one. Generally, you are very welcome, guys. You're very welcome. I am going to finish now because it is 10:15. It's way past my bedtime. I'll be back next Wednesday, obviously. If I can, I'll do the Monday. It just depends, because I want to try and get a few things out. We don't do any tests and they cram the content. When I started module 5. Just interesting. So you've done module 6 before module 5. Actually, module 6 is the one that most students struggle with the most. So that's not a bad thing that your teachers have gone there first than the module 5. Module 5 kind of makes more sense. And also a lot of students, because module 5's in paper 1, they do more revision for module 5. And so therefore, having it close to that exam actually makes some sense for some students. But yeah, If you haven't done, please do check out my videos on my channel. Check out my study guides. I am making them as I go, trying to get as much as I can in terms of what topic students perform worst at. I'd say it's not easy to say which topic they perform worst at. I'd say the genetic side of things, they do tend to struggle with. So the module 6 in general is a hard topic area. But different students have different things. I'd say 6.1.1 and 6.1.3. I think it helped in having a greater amount of understanding. That's cool. It does work like that. Right, guys, I am gonna go. I need to finish. I need to do a few things before I can actually go to bed as well for my group tuition sessions that I had tonight. So thank you for joining me. Thank you for sharing, liking my resources. I do appreciate it. And I hope that it does